I'm going to be presenting in English, if you don't mind, so that we can then reuse this video afterward. So first, thanks for granting me this platform to present the activity of both our companies, Cynbiose and Cynbiose Respiratory. So my name is Joachim Confet, and I'm the Chief Scientific Officer of these companies. So first, Cynbiose. We are a contract research organization created in 2008. We are based to the west of Lyon. And we are a preclinical CRO specialized in non-human primate models, purely the in vivo part of the studies. The type of service that we offer are mainly preclinical exploratory studies, non-regulatory, and also customized proof of concept models of human pathologies in non-human primates. Our business model is mainly fee-for-service, but we are always involved in research and development programs, both together with other French entities, but also internationally. Regarding our facility, we are able to handle BSL-1, like classical preclinical studies, up to BSL-3 using infectious pathogens. And we are currently approved to handle GMOs up to BSL-2 in large animals. We are not GLP accredited, but our whole quality management system follows strictly the GLP principles with a dedicated full-time quality system, quality assurance manager. Regarding animal welfare, we are accredited by the ALAC since 2015, meaning that we go above and beyond what is expected from a regulatory standpoint regarding animal welfare for our non-human primates. So we are currently housed in the campus of the veterinary school west of Lyon, and we can handle up to 100 non-human primates. But thanks to a PA3 grant that we received end of last year, we plan to build our own state-of-the-art facility by 2022, which will enable us to about double this capacity. Regarding Cynbiose Respiratory, which is a subsidiary of Cynbiose and an offshoot or spin-off of the Research Center for Respiratory Diseases, an INSERM lab based in Tours, this company was incorporated in 2017. And contrary to Cynbiose, which is specialized in non-human primates and in vivo studies, but is, I would say, generalist in terms of scientific topics it can address, Cynbiose Respiratory can handle both in vitro and in vivo part of the preclinical studies in non-human primates, but also in rodents. The main thing being that their focus is on respiratory diseases and the assessment of the toxicity and biodistribution of aerosol therapies. The offer, again, is mainly fee-for-services with a constant R&D programs aiming to improve our offer. So the type of services cover a very wide range of preclinical aspects, but the type of studies can be divided mainly in three categories. So I would say first, the pharmacology studies, which cover pharmacokinetics, biodistribution, pharmacodynamics in non-human primates. The other type of studies would be what we call early safety, or basically non-regulatory, non-GLP safety studies. For example, dose range finding or other non-regulatory exploratory studies. I would say that mainly most of the protocols we design for our sponsors are hybrid designs with elements of PK and also of safety in order to maximize the added value of each animal, but also to be cost efficient to our sponsors. So these sponsors, by the way, are mainly cover the whole range of the pharmacology industry, from startups, biotechs, big pharmas, but also academic research teams from France, but also internationally. The main type of products that we will test in these exploratory studies are, since we work in non-human primates, mostly biologics, immunotherapies, lots of oncology, of course, also autoimmune diseases, lots of vaccines, because these species, the macaques, are very relevant for this type of drugs. We've been tasked recently to handle lots of gene therapy studies, which entails quite a lot of challenges, I would say, regarding the screening of the animals in terms of their serotypes and their sensitivity to this or that AAV subtypes, or the ability to handle large animals infected with GMOs, which can be quite challenging in terms of biosafety. We also test a lot of small molecules, and we have accumulated quite an expertise, for example, in the drugs targeting the central nervous system. First, in terms of toxicity, because we have access to a state-of-the-art imaging facility with MRI, PET MRI, CT scanner, etc. And we have developed our own evaluation grids regarding the behavior of the animals and also cognitive motor tasks that we can train the animals on in order to specifically spot and grade behavioral deficits. And also on the biodistribution side, since after administrating or dosing the animals with a drug, which can be classic IV or peros, intranasal, anything, can also go through a very specific route of administration, like intrathecal or intraventricular, this type of administration. Then we can perform sequential CSF sampling in order to have what we call a central PK that we can compare to plasmatic data. If the sponsor needs to go more into details regarding the specific concentration of its drug or a specific metabolite or even a neurotransmitter in a specific part of the brain, we can also perform brain microdialysis studies in order to specifically dose these items locally. And I would say, scientifically speaking, we have already directed 2 PhD theses in central nervous system, and myself, before joining the company, have a 10 years career in neuroscience in non-human primates. The last type of studies that we provide are what we call efficacy studies or proof of concept, meaning that we will either develop de novo a new non-human primates model of human pathologies, or we will implement the one that we already published and validated. So the one that we already developed or can always be consulted on our brand new website, but cover the whole range of relevant physiological functions. For example, the central nervous system, since we have very recently published ischemic stroke model in non-human primates, inflammatory-related diseases or processes like the skin graft rejection in non-human primates, cardiovascular diseases like heart failure, respiratory diseases, or I would say bone and muscular disorders like cartilage defects or traumatic osteoarthritis. And of course, we can always develop new models if the primate as a species is deemed relevant. Since I only have 20 minutes, I'm going to be focusing mainly on 2 points regarding our expertise. And the first one is the ability to handle human pathogens. So, as I was saying, our facility allows us to handle pathogens from a BSL-1 to a BSL-3 biosafety level. And this expertise has been with us, I would say, since the beginning of the company, since the CEO and founder of Cynbiose, Hugues Contermain, which is also one of the co-founders of the AFSSI. Before starting the company, had a long career at the Institut Pasteur and then at the P4 laboratory in Lyon, where he spent his career developing animal models of human pathogen infections. So currently, what we can provide is a catalog of non-human primate models of tropical infections and innovative and very well characterized models of respiratory viral infections. For example, the respiratory syncytial virus or the metapneumovirus in non-human primates. We can also provide rodent models of acute and chronic bacterial and viral infections or even co-infections. These models, since we have right now an expertise to handle, I would say, infectious models at large in this species, can be extended depending on the request of the sponsors. Typically, the respiratory viral infections requires very specific sets of skills, like the ability to perform bronchoalveolar lavages, or the ability to perform very deep swabs or lung biopsies, etc., or even lung microdialysis if needed. So these models or the results are either ongoing or collaborative, ongoing or past collaborative R&D projects. And for example, right now, it's very much an actual topic. We are characterizing COVID-19 model of infections in the African green monkey. And we are also involved in the development of a sepsis model in the non-human primates, again, cynomolgus macaques, with an acute phase and a recovery. So in these infectious models, of course, we characterize the interaction between the pathogen and the host, but we don't stop there because we believe that as a preclinical model, the animal should not be considered isolated from the whole microbial ecosystem that it harbors in its lungs, in its guts, for example, on its skins. And that's why we also characterize the microbiota of these animals, for example, in case of respiratory syncytial virus in the lungs. But for example, for coronavirus, we'll also target what happens in the guts. And thanks to this, I would say, this focus on the interaction between the microbial environment and the viral pathogens, we launched the Cynbiome project, which is the first preclinical network of excellence for the investigation of microbiota and infectious diseases, mostly in non-human primate models. This project involves the building of a new facility in the next 2 years that will allow us to tackle these very challenging topics on a large scale, and has been made possible by a PIATRA grant that we got in 2019. The next topic I would like to address is the specificity of Cynbiose Respiratory, meaning the ability to assess the biodistribution and the toxicity of aerosol therapies. So why aerosol therapies, right? Because this field is at the same time full of promises and of challenges. Promises because it allows to directly deposit the drug at the supposed site of actions, which would be the lungs, while minimizing the overall exposure of the body. But it's also ripe with challenges because to nebulize a drug is no trivial matter. There is lots of formulation topics to be addressed. For example, the stress due to the air-liquid interaction or the potential folding of the peptides if what you want to nebulize is biologics, etc. And that is why the type of tools that has to be used in order to properly assess these topics is both from in vitro and the in vivo side of the preclinical investigation. So at Cynbiose Respiratory, what we can do is first assess the physical properties of the nebulized process. So meaning we have to work on the formulation itself and the combination between the formulation, the drug formulation, and the device, because the nebulizing process can rely on lots of different devices, and what is to be tested is the combination between the two. So these tests are based on the American and European pharmacopoeia. And once this step is cleared, we can move on to what we call deposition studies, meaning that we can mimic what happened in vivo using 3D printed casts of either macaques, which will be our target species when we do the in vivo study, but also newborn humans. We will use these casts in order to mimic the deposition of the drug in the upper airways, but also in the lungs if needed, in order to precisely characterize the different parameters of the nebulization process, the size of the puffs, the angle, or the depth of insertion in the nostrils of the nebulizing device, etc., etc. So once this, I would say, screening part of the study is done, we can move to the in vivo part, where we can provide sponsors with, I would say, classic pharmacokinetic studies, where the only difference would be that we can compare a classical route of administration like Peros or IV to nebulization. But then we can also go more in detail by performing pulmonary microdialysis or even scintigraphy studies in order to look at the relative exposure of the lungs compared to other tissues. If we, let's say, bet that these drugs have an efficacy, we can also suppose that they might be toxic. And that's why we can also provide the sponsors with specific pulmonary toxicity studies, meaning non-regulatory, but regular toxicity studies with the added value of respiratory function assays, looking at the compliance, etc., of the lungs before and after nebulization. Another very relevant aspect related to aerosol therapies is what is called nose-to-brain delivery. So the brain is a notoriously hard to reach organ, thanks or because of the blood-brain barrier. But by nebulizing drugs right at the olfactory part of the nasal cavity, one can bypass this BBB by going through an extra paravascular and nervous pathway, which is not completely understood right now, but allows to maximize the exposure of the brain while having basically null plasmatic concentration. So to do that, we will first nebulize the animals using the device that we characterized in vitro before, and we can then sample at the same time the blood of the animals, but also their CSF, or we can also combine it with a brain microdialysis. Again, if we assume that there might be efficacy, there could also be toxicity, and we can, as I described before, have access to a whole range of assays aiming to characterize the potential neurotoxicity, or I would say behavioral deficits of the animals following the dosing of these specific drugs. So if I have to summarize, and there are 5 points that I would like you to remember concerning our 2 companies. We are here to de-risk your lead candidates and maximize the chance of success of the INDs by using a very much relevant species, which is the non-human primates. We can offer you scientific and technical advices on our 2 fields of expertise, which are aerosol therapies and non-human primates. We are able to design very complex protocols, giving you high added value data, but constrained in a very robust QMS following the GLP principles. We have an experienced team with more than 12 years of existence right now, with a lot of flexibility and agility. And most of all, we retry and strive to involve the sponsor every step of the way during a study, during the design of the protocol, during the protocol with a very frequent reporting, and then going hand in hand with him to or her to interpret and analyze the data. So, and this said, I thank you for your attention and can answer any question you might have.